Hey everybody, welcome back to Research Methods and Experimental Design in Environmental Studies. We're talking about experimental design today. This is going to be a lecture that we do in class, and I'm also going to go ahead and reproduce it here for you just so that you have an option to go back and see this content multiple times. This is a pretty important one, obviously, since it's kind of the basis of the whole class. Um, Keep in mind that we're learning our statistics in R and also just statistical theory in class as well, so that we are able to actually design experiments and so that we can read and understand and interpret experiments that other people have designed. It's all of these components together that allows you to make a really good experiment. Um, so we're going to be working through a lot of stuff today. This is probably going to be a long one, but we again are going to see all of this in class as well. So if you're watching this before class, we are going to get it in class, and then we're also going to have a chance to work through some case studies in class in groups, which is going to be hopefully really fun. So let's talk about experimental design a little bit. The first thing that I want to tell you about experimental design is that this is your chance to flex your interdisciplinary muscles. Experimental design is an art form as much as it is a science. It's a creative process where we're looking to do creative problem solving and work within the limitations of our materials, our facilities, our time, and also all the people that we have. If you're working on your own in a remote area, you can only do a certain kind of experiment. If you're working with a huge team in a really well laid out place with lots of resources, you can do a totally different kind of experiment. So you want to think about what you have at your disposal and take advantage of what you do have and work around your limitations. So that requires some really creative problem solving. I can give you plenty of examples, please ask. There's also a lot of possible designs for just about any kind of experiment, and they all have trade-offs. And this is where we get to maybe the most important piece of information I can give you about experimental design. All experiments have design flaws. There is no such thing as the perfect experiment. We can draw it out on paper as a perfect experiment. There's probably still biases and flaws, but we can try our very best to do the very best that we can. But flaws are inherent in the system. We're going to make mistakes. Problems are going to occur. There's going to be flaws. We just need to acknowledge them and try to deal with them as we go through the process. We want to try to minimize them, and we also need to figure out how to analyze our data to either deal with the flaws or find a way to make the flaws at least known in the writing. We must try to interpret our data within the context of our flaws. Here's an example of an experimental design setup where you have two different treatments, the black treatment and the white treatment. And you can see that we have experimental units, which we can just say they're experimental units if you want. If you want to think about what this actually is, you could picture this is 8 tanks laid out in a row, and 4 of them are connected to one source of water that is the black source of water. Let's say that's hot, and the white source of water is a cold treatment. So we have a hot treatment and a cold treatment. We've got these 4 experimental replicate units, or 4 tanks each, and they're kind of randomly dispersed across, let's say, a shelf. If you've been to my basement lab in Carnegie B15, yeah, B15, maybe you have a sense for what this might look like. But that's an example. So there are problems with this. It's not perfect. There's a lot of things we want to think about. And we're going to try to unpack how to look at these diagrams, how to make them, how to think about them, and really figure out like what's good, what isn't good, and what are our limitations. So that's where we're working towards today. But first, we're going to have a little bit of fun. Let's talk about why experiments are flawed. This isn't me, but this has been me on more than one occasion. This is a person who is on a reef rubble site adjacent to a coral reef, and they are trying to, via scuba diving, do an underwater transect, and they've laid their transect tape, and they've got their PVC quadrat laid out right there. You can see that little blue bag that's full of some of their other gear. They've got their slate in their hand, and they are currently just being pushed back and forth by the surge. The water is shallow, the waves are crashing in one direction, and the surge is pushing them all over the place. So unless they're holding on for dear life and also writing and also moving their pole and their PVC pipe to measure everything, they really can't do it. So, you set out with this plan to lay out this really nice, neat grid of transect tapes. And you go through your grid of transect tapes with your grid of quadrats and just like do a perfect analysis. Maybe you've done this in environmental science classes here. It's a lot easier in a terrestrial forest, or even in the Rocky Intertidal than it is fully underwater on scuba. The truth of it is the ecosystem doesn't really care about what you're trying to do. So, this brings back memories that mostly are stressful and sad for me, but also it's very funny. We can try our best. We're just not always going to get there. So experiments are inherently flawed because basically ecosystems and biology, they don't care about your hypothesis. So for example, here's a paper from a colleague of mine that I really, really like that has suffered a little bit from something that we call in the field demonic intrusion. Demonic intrusion is kind of just a funny term to say an unexpected circumstance. And in this case, the unexpected circumstance is an octopus. As maybe you know, octopi are pretty smart. Here's an octopus using a coconut shell as a defense mechanism and also for mobility. In this case, this paper by Kenkel et al. Is about corals that they're collecting, cutting into small pieces, and then transplanting all over a reef. And they set out at the very beginning of the experiment to collect 30 fragments from all of their sites. So here's a table. Yes, I forgot my little proverb. The road to hell is paved with good intentions. We all meant well, and then things got weird. That's our demonic intrusion. So we have these 4 regions, the lower keys and the middle keys, and these inshore and offshore transplant sites. And we can see that the initial N at the beginning of the experiment was 30. So they put 30 fragments in all these different places at the beginning of the experiment. They came back 6 months later and there was mortality. They collected fragments at 6 months. So they got 15, 5, 10, and 13. And they collected at 1 year, another 15, 6, 5, and 5. So if they had had no loss of corals at each site, they would have come up with total N loss of 0, and they would have collected 15 and 15. They didn't collect 15 and 15 at any sites except for this inshore lower keys site. So there was mortality, or there were other issues where the corals were lost somehow. But you can see that they've got the total N loss of 19 in the middle keys. And the reason for that is because when they labeled their corals so they could understand who was who underwater, they labeled them with livestock tags that were shiny. So they're metal livestock tags that they had stuck next to the coral or attached to the coral with a clip. The octopus thought that was pretty cool. So the octopus was swimming along and saw these shinies and was like, oh, shiny, and it hooked them. So when they went back, I believe at the 6-month time point to collect at the Middle Keys site, they were diving on the site, and these are colleagues and friends of mine, and they told me this story at a conference. They were diving at the site and they came upon a pile of their tags, and they were like, oh, maybe this will explain where our corals went, because they'd all gone missing. And it was like, kind of close-ish to the site. And they stuck their hand in to get the tags, and the octopus just like shot an arm up and was like, these are my shinies. So they lost all of their samples. Sometimes it happens. Even if you do like the perfect setup, this happens. I've done something similar myself, where I've done some underwater coral restoration and transplantation. And we zip tied a bunch of our corals down to like an underwater steel table that our colleagues had put in for us. It's pretty sturdy. But over time with storms and things like that, the zip ties can break, or like the things that we had attached the corals to were like smashing up and down with the current, and they just broke off and went away. In some cases, the super glue we had used to adhere them to the surface had actually stopped functioning and the coral was just gone. So the thing we put the coral on was still zip tied to the table, but the coral was just gone. And we used the same super glue on every coral. We did exactly the same thing every time, but for like 1 or 2 of them, it just didn't work. These things happen. Don't forget also, when you're doing biological replicates, that things can just die. That happens all the time. So just so you've seen one of these before, I'm showing you an experimental diagram. And I know that this is confusing, and this isn't necessarily the best place to start because this is a relatively confusing paper, but here is what this diagram looks like. It's from the paper. You are going to be seeing diagrams like this from papers. Here they've got a map of Florida, in case you aren't familiar. So they've got Florida here, And they've zoomed in to this little box, which is the southern tip of Florida. You can see the Florida Keys here. And as we go up, the Florida Keys Marine Sanctuary area is this bit. And then the Dry Tortugas is out over here. So they're showing you now that they're sampling inside of A, which is their lower Keys area, and B, their middle Keys area. So they've got these nice blow ups to show us that. And then A and B are these really detailed maps of the sites. So they're showing you with a color scheme, the inshore site is in orange and the offshore site is in blue in A. And they transplanted corals both from an inshore site to another inshore site. So that's their in and along or in novel treatment. They also cross-transplanted from inshore to offshore at the exact same latitude. And then they also did an off novel or an offshore novel from offshore to offshore. So they're transplanting between inshore and offshore to see if there's an effective inshore and offshore. And they're also transplanting from an offshore to another offshore and from an inshore to another inshore. So they're looking to see, is the variation within inshores and offshores bigger? Is the effect of the variation between one inshore site and another inshore site bigger than the variation of transplanting from inshore to offshore? So this is actually really, really good experimental design. And if their sample sizes had worked out perfectly, this paper would have been really, really, really high impact. Instead, it's still in the Journal of Ecology, which is pretty good. So they did the same thing in the offshore environment. Or I guess, they did this. This is what they did in the lower Keys, inshore and offshore, really clear. In the middle Keys, inshore and offshore, a little bit less easy to understand, but they still did like an inshore in pink and an offshore in green. The channel is just at a different angle. So that's the experiment. And then you can see here, they took 5 fragments from each individual and they fragmented them off into 15 frags that they put all over the reef. So, and then they collected in at different intervals. You're going to see this again. I just wanted you to see it and see if it makes sense. Okay, so I'm thinking of the idea of demonic intrusion, which is why you've got the cover for the book Good Omens, which is a pretty funny parody of a lot of things. If you haven't experienced that, there's also a Netflix show. It's funny. So we're going for this idea of quick and dirty versus precise and accurate. Precise and accurate, I also will refer back to this title, The Nice and Accurate Prophecies of Agnes Nutter, which if you read it, you get it. It's fine. You can make fun of me for being a nerd. So our goal here is to figure out how to thread the needle. Are we going to be quick and dirty? Or are we going to be precise and accurate? It's the great conundrum of our scientific time. How do we collect the amount of data that we need in the time that we have using what is always limited resources that we have at our disposal? How do we find the perfect match of conditions that allow us to do the best possible experiment that answers our question or deals with our hypothesis? We might try this framework, the 9 rules for experimental design from Sahadi 2016. Here are Sahadi's rules. Number 1, have clear hypotheses and predictions. Write down your hypotheses, write down your predictions. So that means you should be writing down null hypotheses and potential alternative hypotheses. Think about what could happen and why, right? We have to use our own knowledge of the system to come up with a good hypothesis, but we also might have some predictions for what would happen. We should always be thinking about those things and be looking for them in our experiments. Second, we should ensure that our sample units are independent of our samples. So basically, we just need to have independence. Does one sample depend upon the next? If so, maybe that's a problem. Does our unit of sampling depend upon our samples? If so, that's a problem. So what are we measuring? We're measuring growth rate. We're measuring the height of our shell, something like that. Is one shell height independent of another shell height and of another shell height? It would be if we measured across multiple individuals who were not interacting. So we need to be thinking about independence, and we can talk more about that in class. We also want to specify our controls, and the paper didn't say this. We also need to have controls. The number of experiments that have no control and make statements is too many. It's way too many. There should be a control of some kind, where we either don't change things or we keep things the same as what is in the natural environment. We're either holding variables constant or we're basically just looking at a natural ecosystem. You have to have some kind of control, and it's not as hard as you think to come up with ideas for controls. People should always have them. We also really want to have a random sample or random samples in our populations when we're doing this kind of work. So we want to be thinking about how we're sampling. We've already learned all about that, trying to go for randomness or as best we can, right? We also want to have replicates. the law of large numbers, or when in doubt, increase your sample size, right? So we want to have many random samples of our population so that we can get closer and closer to the true population estimates, right? We want to try to have a small amount of standard error or standard deviation around our sample mean and hope that our sample mean is precise and represents our population. We want to avoid this thing called pseudo replication when possible. We are going to dig into pseudo replication probably ad nauseum today. We try to not have it. It's really hard to not have it, especially in experimental systems with water. So that seems relevant to what I do. So we're going to talk about that. We also really want to use appropriate statistical analyses. I will go back to that thing I said about controls. The number of people who don't do controls correctly is too many. The number of people who don't understand or don't do the right statistical analyses is also too many. When you come up with a hypothesis, you should probably be able to say, here's the stats I can run. And if you have a hypothesis that you don't know how to analyze appropriately, you probably want to rethink your hypothesis and reframe it into a statistical framework that makes sense to you. There is no shame in using simple statistics to answer questions. That is fine. It's better for you to understand what you're doing and why than it is to do something complicated that you don't get, because you have to explain it to other people. And if you don't believe in you, nobody else is going to believe in you either, right? So think about the stats you know, and let's use them. And if you really want to learn a new stat to solve a problem, that's actually pretty good impetus to start learning stats. The reason that I learned statistics, that I learned programming at all, was because I had questions that I couldn't answer. So I learned how to answer. It's what drove me forward. Maybe it'll help you. 8, do not base the entire study and interpretations on p-values alone. You've probably heard that p-values are squishy, that p-values are fake, that p-values are just totally bullshit. Like, all of those things are at least some flavor of true. We are going to dig into that over the course of the semester. I'm happy to talk to you about it now or later, depending on your interests or your need. But stats are a tool. They are an imperfect tool, and the p-value is not the be-all and all. Things can be significant or relevant without having a p-value less than 0.05. And things that have a p-value less than 0.05 may not matter at all. You really have to have some interpretive knowledge. And we are going to build that. And then finally, you got to be innovative and insightful to make a good experiment. It's just true. So you approach these rules and you try to be creative, and you do the best you can. Really importantly, these rules are basically like tenets for checking off that you're doing a good job. So if you can demonstrate that you understand how to do these things to me, you're going to succeed in this class. You're also going to succeed in our experimental design project, which we'll talk about in class. Okay, those are our rules for experimental design. On to the next. Because I'm an ecologist, we are going to talk through the types of experiments with an ecological or at least a science lens, so a natural science lens. And I'm going to use types of ecological experiment here just because that's what I think of. These are not the only kinds of experiment. These are the most common kinds of experiment in my field of expertise. There's definitely other stuff with human subjects, different surveys, things like that. Natural sciences are different than humanities and social sciences. That's okay. I study natural sciences. I study things without backbones, partially because it takes a lot of paperwork and permissions to study things with backbones. Just recognize that your design needs and interests will vary based on the field or the subfield that you're after. But there are still lots of different kinds of experiment, and these are all generally some examples. We'll start on the left. This is the first kind of experiment that natural scientists did: field surveys and observational studies. We went out into nature and we looked at it. We sat at night and watched the stars and observed. We took notes. We went out into a field, we went out into a forest, and we just looked. We looked for birds, we looked for invertebrates, we looked for whatever was there, and we wrote down what we saw. Then we went somewhere else and we wrote down what we saw. Eventually, we started putting out grids and measuring how much we saw, where, and when, and why. That is field and observational studies. You go out, you look around, you quantify what is there. You don't really manipulate, you more observe. So, for example, the kind of work that I do sometimes in the intertidal is to move around across the Gulf of Maine to go to similar kinds of ecosystems with similar rocky intertidal ecosystem like structures and quantify what organisms exist where and how much. Is that changing over time? That's something that we're going to be setting up here at Bates, trying to build a long-term study to understand how the Gulf of Maine is changing with climate change. Obviously, other people are doing that as well. So when we work together on it, we can really build this nice, robust data set across the entire area of the Gulf of Maine. So that it still has a lot of inherent value. This kind of experiment is often hard to fund because funding agencies don't necessarily love this kind of science, which they call monitoring, because it doesn't like push the envelope with technology or anything like that. But we live in unprecedented times. The world is changing rapidly. We do not understand it. A lot of the marine ecosystems that we're looking at, we never understood before it got too hot. So we don't have a lot of sense for what's happening. And if we don't do this field and observational work, we won't know what's possible, what's changing where, when, and why. And then we can't respond to it. So we need to be out in the field looking and using the science that we gather from the field to make decisions. So funding agencies and non-governmental agencies and state governments and federal governments, they should care a lot about this kind of work. But when it comes to money from grant agencies, it's sometimes hard. So a little bit of a soapbox for the benefits of this kind of work, partially because it's what I do, but it's because I really believe in it. It's very important. So we have that kind of study where we're out looking around. We also have a manipulative field experiment where we might go out into the field and make something different, change something. Maybe we'll go out into a salt marsh and we'll say, what happens if I remove all of the predators, all the predatory, say, whelks, or all of the grazers, even, let's do that. We're in a salt marsh, it's mostly grasses, and the number one grazer is a particular kind of snail. What if I build a fence around a 4 foot by 4 foot plot of salt marsh that's like 6 foot tall, but is made out of mesh so the light can still come in so that my salt marsh grasses can grow? What'll happen to that plot if I go into it? This is a thing people really do. You make these 4 foot by 4 foot boxes so that nothing can enter. These snails can't enter because the mesh is too small, and you make it vertically high enough so that they had to put in a lot of energy to get up and over. They won't. And then you go inside of the box after you make it and you pull out every single snail. If you go into salt marsh ecology or intertidal ecology, someday this will be you. This could be you this semester or next semester if you want. It's a lot of work, but you make these boxes, you make a bunch of them, high replicate, right? And you go in and you pull all the snails. You let it run for a couple of weeks, a month, a couple of months, whatever you want to do. And then you go back and you count how many snails are in there, how fast is stuff grown relative to plots where we didn't do exclusion, things like that. So is there an effect of these grazers on the growth rate of these organisms or on the other stuff that settles onto these grasses, or just the overall makeup of the ecology of the system, like the different kinds of grass that exist? And we can also do something like this in the field. So those manipulative experiments where you're taking corals from one place and moving them around, that's also a manipulative experiment. What it does is it allows you to see how things respond to new conditions in the wild. So you don't have control so easily. It's a lot harder to control all of the many variables that happen in the natural environment, but you're able to do a really interesting experiment where you can move things, you can manipulate some variables and see how that change makes the organism respond differently relative to if you had just left that thing alone. So it's really powerful, but it often makes us have more questions than answers when we're done. We then go, why? I moved you from your favorite home with this beautiful clear water where the water temperature was perfect and all of your friends were there. And I moved you to this really nasty, swampy, sediment heavy, sewage infused river mouth. And you were like, this is neat. And you did better. You grew faster. Like my first question when I did this happened to me. Why are you like this? Like what made you do that thing? I want to know. You collect some samples, you try to do some molecular genetics, you bring them back to the lab if you can, and then you try to really crank on finding those specific mechanisms. And that's where our third component, the manipulative lab or mesocosm experiment, comes in. Once you identify some really interesting responses in the wild, you can isolate variables one by one or 1, 2, or 3 at a time to see which one of those variables drove my response. And if I can see that response again, maybe I can even quantify, like, mechanistically within an organism, how did that happen? Why did temperature affect the growth rate? Why did sediment affect the growth rate? It's pretty awesome. So that is my long-winded explanation of these 3 kinds of experiment. I'm hoping that in class we can talk through this concept. What are some examples of each of these things? What are some disadvantages and advantages of each? let's think about them and kind of compare and contrast and just understand like when would we use one versus when would we use another. And there's so much more we can unpack there. So hopefully we'll talk about that in class. Let's jump into the requisites for an experiment. These are the things you've got to have. This is kind of like those 9 that we just looked at, only it's like much more specific. And this comes from Carbon 2014. You got to have appropriate controls or else you're wrong. You have to have meaningful treatments or else what you're measuring doesn't matter. So when you think about your hypothesis, and then you think about what treatments you're going to have, can those treatments address your hypothesis? If that's not true in tangible ways, then something's wrong and you got to rethink. You need replication as well. How many replicates can you get? The answer should be as many as you can possibly handle, within reason, of course. If you're going to grow your experiment too big and you can't take care of it and it'll all die, that's not good. You want to be able to do something manageable, but also have high enough replication that it'll matter. And then you want randomization and interspersion of treatments. And that's something we haven't talked about yet. The reason we haven't talked about it is because we have to unpack some problematic history of the field to do so. So here we go with that. This is going to be sort of like a podcast aside where we go down a little bit of a rabbit hole, but here we go. There's this classic paper in experimental design, came out in 1984 in Ecological Monographs by Stuart Hurlburt, who is no longer at San Diego State. This was a long time ago. I believe he's retired. Pretty sure he's retired. But it's called Pseudoreplication in the Design of Ecological Field Experiments. This paper was really, really, really important. It changed the way people like use different words, and like some of the designs we've seen have come from this paper. There's a lot of good things in this paper. It's really important to point out that sometimes scientists who are also people are pretty flawed. This guy, kind of the worst. So I try not to teach exactly his stuff, but just recognize that you will see this word pseudoreplication. It does come from this paper from 1984. We are going to talk about the components of experimental design that come from this paper without really like digging into specifically this particular paper. There's lots of other ways that we can talk about this. So let's just move on from that and recognize that like we're going to talk through experimental design and this concept of pseudoreplication, and then it comes from this relatively recent paper in 1984. So see also the debate. This paper from Jeff Schenk and Thomas Kennel shows that they think pseudoreplication is a pseudo problem, suggesting that maybe it's not that big of a deal. There's another editorial that just came out this year in Ecology Letters that said called The Principles of Experimental Design for Ecology and Evolution by Dustin Marshall. We're going to read that one and we're going to talk about it in our next class period. Or actually, we already talked about it last class period. We'll see which one of those is true. It's going to depend on timing. But in that, Dustin Marshall says why he prefers not to use the term pseudoreplicates, but uses something called subsample instead. And the reason that he uses subsample is just that it's a simpler word. Pseudoreplicate is a little bit gatekeepy. Subsample seems like a sample of a sample. That sounds like it might be a problem. It's a little bit easier to understand. Whereas pseudoreplication is kind of like, what does that actually mean? Like you're kind of replicating? I don't know. Like it's a bit of a word salad. So we're going to dig into some of these concepts and look at some experimental designs. But first we need to take a step back and really talk like define some terms. So we've been talking a lot about this thing called control. You got to have it, right? So what is it? It's treatments against which manipulations are compared. So like the treatment of no treatment is a control. The experimental unit, which we didn't define earlier, is the smallest unit to which a treatment is applied. If I have 4 tanks that are hot and 4 tanks that are cold, each tank is a treatment, question mark. Or if I have 20 organisms inside of each of my tanks, is each organism an experimental unit? We're going to dig into that. It's actually the tanks. The tanks are the units. If we consider each individual to be a unit, that's subsampling or pseudoreplication. So we're going to get there. And then finally, meaningful treatments. We often change things, manipulate variables, and we need to be careful about how we set up our treatments to make sure our treatments actually do something. They actually test the hypothesis. If not, why are we here? Now, we can finally talk a little bit about replication. We must replicate independent experimental units across all treatments. Why do we need to do that? hopefully you'll recall from the last couple of lectures we've done, which have now been a couple of weeks, it's really important for distributions. If we want to approximate in a normal distribution the true mean of a population, we need lots of independent replicates from our population or of our samples so that we can get a nice bell curve. So it allows us to quantify random error due to unknown factors. That's why we want replicates. What are the chances that when I take a sample, that sample isn't representative of the full population, but is representative of some weird thing that happened one time? If I take enough samples, I can get rid of some of that noise. Here's an interesting graph that we are going to actually look at in class and talk about some more. Hello? Okay. Does alewife length vary by location? In class, I'm just going to hand you this and say, answer my question. Talk to your friends. What's the answer? Does alewife length vary by location? Look at all 4 graphs and try to explain to me what the answer is. Good luck. Be specific. I'll leave that for class. I'm not going to spoil it, because if I spoil it here and you watch ahead of time like I'm asking you to, you're going to spoil it for everybody else, and that's no fun. So just keep in mind what mean, range, standard deviation, and standard error mean and how they're different. Okay. Finally, we can talk a little bit about bias, and then we're going to start looking at different experimental design diagrams. How do you prevent bias from unknown factors that could lead you to false conclusions? We don't want to have false conclusions, false positives, false negatives. I feel like that's the kind of terminology that's in our heads now since 2020, because a lot of COVID testing has these false positives or these false negative rates. How can we know what we don't know? We can't anticipate everything. Sometimes we just have to try to build the best experiment that limits our biases. And then we can talk about our biases and our methods and in the discussion when we write the paper. Hey, we have these shortcomings, but we still see these things, or we have this really clear difference, but there are some variables that we couldn't quite quantify, or there are some issues with our experiment. So we just want to think about that all the time. All right, let's talk about this idea of randomization and interspersion and show some exact experimental diagrams. This is a big diagram. It's going to be okay. We're going to work through this. We're going to talk about these things, but I just want to show you what this means. We said already that replicates have to be independent and unbiased. So let's just assume all of these little boxes, each one is a replicate. Let's assume that they are all independent and unbiased. Here are some different experimental designs. We have at the top this completely randomized design. There's 2 different versions of it. The first is just a line where we're seeing we have 2 treatments, black and white, and we have randomly each time, basically, let's say we're setting up our experimental plots, and we have to decide which treatment to apply. And we're in the forest, and we've just laid out 8 grids, 8 transects. And now we're ready to either put fertilizer in them or not. The way we would randomize it is we would bring a coin and we'd flip it and say, heads, we fertilize, tails, we don't. And you just flip it every time. And you had to have 4 of each. So once you get 4 of the same thing, then you know you only have the other one left. But in this case, right, we just randomly flip, we get black, then we flip, we get white, then black, then black, then white, then white, then black, then white, right? Random. You can also use a random number generator. You can write down a random list. You can have somebody else give you pick a number between whatever and whatever. That's completely randomized. If you set up your plot instead of in a straight line in a grid, which is probably what more people would do, you basically play Sudoku with it. So you want to make sure you have going left to right, 2 of each treatment, and going top to bottom, 2 of each treatment, except for they don't do that. So they don't even follow their own rules. So apparently it's from going left to right, you have to have 2 of each treatment in each line, in each row. So they've got 2 of each treatment in this row, 2 of each treatment in this row, 2 of each in this row, 2 of each in this row. And they apparently don't care about the other rows or the columns. That was their decision. Not how I would do it. But you're going to see an example of maybe how I would do it. Maybe it's more systemic. So randomization going across the column, and you have to have randomization going left to right as well, as long as you have 2 and 2. So hopefully that makes sense. We're going to look at these in class as well. That's your completely randomized. You can also, first of all, like, I want you to think about what are some problems with that. why might that not be ideal? If you're setting up a grid across space, why would random not necessarily be ideal? Think about that. Sometimes it's not, and here's why you would use the randomized block, because it's not ideal. In a randomized block, there are units. We have 8 replicates in our horizontal line here, and we've got them divided into groups of 2. And we're saying in each group of 2, you got to have a black and a white, right? So one of each treatment: black treatment, white treatment, white treatment, black treatment, white treatment, black treatment, black treatment, white treatment. They're black and white simply because this paper, this diagram comes from before color printing. So those are the options are like fill in the circle and leave the circle open. Just in case you were wondering. So we've got B1, 2, 3, and 4. And inside of each of the Bs, we have both one of each treatment. So again, you do a coin flip for the first one, and you get that one, say it's heads, therefore it's black, which was fertilized, and then the other one has to be tails. And then the second time we flip tails, therefore no fertilizer, so the other one has to be fertilized, so on and so forth. Now you can also do randomized block by making blocks inside of your grid. So they're making 4 blocks: block 1, block 2, block 3, and block 4 across the horizontal axis or across the rows. So each row is a block, and they're saying you got to have 2 of each in each block. So they made a grid where you had to have 2 of each in each block. They, again, don't have it going vertically in columns. And they apparently also don't have that in this top one. I guess I just made that up. They literally just flipped a coin every time. And it just came out that there were 2 and 2 each time. So I think I explained that wrong. And that happens. It's completely randomized, which means every single square, they just flip the coin. And it was either white or dark. Okay. And down here, it's either white or dark. But there has to be 2 white and 2 dark, or I guess 2 open and 2 closed. I don't know. However you want to think about that. Filled in versus not filled in squares. There has to be 2 of each in each row. That's randomized block. So it's randomized within the block, but there are blocks that force us to have a little bit of structure, and that might be good. So compare that to randomized, completely randomized, where there is no structure. Why might randomized block be more ideal in some situations? See if you can come up with an idea for that. Lastly, maybe you're like, this isn't organized enough. I want it to be neater. I need it to be neater. My brain requires that. I understand. I get it. That's how I am too. There's this thing called systemic layouts, in which we say there is a pattern. We will follow the pattern. This is really great for a lot of reasons. One of the reasons is that it's easy to remember. If you randomize stuff, you got to keep track of it all the time. If you use a pattern, then you have a pattern, and the pattern is always the pattern, and that's it. I love that. That's easier. When you're training people, when there's lots of people working on an experiment, we go for systemic a lot because there's a pattern. So in this case, they're saying, if you got dark, you got to have white after. If you have white, you got to have dark after. It's just going to go every other. Duh. And in this case, they're also saying we now have row and column blocks, and we're saying you got to have all these different treatments inside of your blocks. So they, in this case, are giving us an example where there's 4 treatments. So they're saying going left to right, you got to have one of each. Going top to bottom, you got to have one of each. It's actually Sudoku. So it's nice and neat, really easy. And that is systemic layout. So think about all 3 of these. Think about like which ones are more work, which ones are less work, which ones might be more ideal for certain situations. Can you think of times where completely randomized, which is the ideal, doesn't work for us, and why? Why would you go for randomized block? Why would you go for systemic? I'll even give you some examples. Okay. So if we're setting up field plots in a salt marsh or in a forest or something like that, I'm going to ask you to compare and contrast using a completely randomized versus a randomized block. And then I'll ask you also, can you come up with an experiment example in a lab setting where we're using this kind of blocking system or this random system? So think about those for class and we'll discuss. Finally, we were already talking about randomization and interspersion. We've already talked all the way through this example. I just went ahead and did it. So I want you as a group, when we get to class, to sit with this and try to explain it. Look at it again, try to explain it to one another. I've already talked you through all of it. The video is already posted, or it's going to be posted along, if you're watching this, you already saw the video. So you can hopefully interpret what I saw, but I want you to try to explain it to each other. What's happening here? And see if you can tell me what kind of design you think it is. I think that would be an interesting exercise for you. And then we're going to put randomization and interspersion aside for a minute, and we're going to talk about this pseudo-replication concept. Pseudo-replication occurs when we collect multiple observations that are not independent of one another. Independence is a key assumption for all of our statistical analyses generally. So having observations that are not independent can be a concern. Simple pseudo-replication means we have one unit in a treatment and one in control, but multiple measurements taken from each. So here's the example that the Hurlburt paper gives. I have plot number 1 with the gray hatches, and I have plot number 2, which is our X plot, and Y plot, which has no hatches. It's just clear in the background, or no color, or whatever, I don't know. And we have this plot, and this plot is our experimental unit. And then within the plot, we're taking 4 measurements. And then in the other plot, we're taking 4 measurements. And then we're comparing the plots to one another. Hurlburt says, no, that's wrong. That's pseudo-replication. That other paper that we read said, no, that's wrong. That's called subsampling. What you're doing is you're measuring the same thing multiple times. And that is each one of those measurements, because we're measuring the same thing, is dependent on all the other measurements within that system, right? Think of it like this. If this is a blade of grass on X and a blade of grass on Y, and I measure some parameter of it 4 different times in 4 different places, like that's not necessarily comparing different individuals. It's comparing the same individual multiple times to the same individual multiple times. That is not quite right. So that's pseudo-replication or subsampling. This can get really contentious and a little bit confusing if you start scaling up to, like, I have a tank full of organisms here and a tank full of organisms here, and each organism is being subjected to the treatment in the tank, and each organism is being subjected to the treatment in this tank. Is there a tank effect? I don't know. It's complicated, but like, does every organism in this tank, are all of them pseudo-replicates, or are they independent? And how do I know? And the answer to that question is, it depends on who you ask, but they're probably subsamples or pseudo-replicates. And is that bad? What do I do about that? And that's something we can keep talking about, of course, over the course of the semester. The best option would be to take if you have 4 individuals, put them all in 4 individual unattached treatments where they can't interact with one another, where there's no way that they are dependent on anybody else. They're just doing their own thing. They're in their own little boxes. If you do that, you're doing it right. So that's this better design, multiple treatment plots, 1 sample from each. It is not always possible to have multiple treatment tanks, to have multiple treatment plots, or to have 30, which is a good large N of each thing. That is always hard. So sometimes we pseudo-replicate. It's not our intention. It does happen with our designs. We want to try to avoid it. But other people will tell you it's not a big deal, right? So remember that pseudo-replication is a pseudo problem. We can look at that paper and talk about it a little bit if people want to. I'm going to open that up for you if you want to do that. So this is simple pseudo-replication. There's also this idea of temporal pseudo-replication that says measurements taken over time from each plot are treated as replicates so they're averaged together to produce a population mean instead of looking at them as a time series. I'm going to skip through this pretty quickly because I'm not that worried about it. But if I'm measuring the growth rate of an organism over time, or if I'm measuring the length of a leaf in a plot over time, and I measure that leaf once, and then I go back 6 months later and I measure it again, and then I measure it again, and then I take the average and say the size of the leaf is this mean plus or minus this error, that's obviously bad. I think we can all see that that is not correct, because over time the leaf is going to grow or die, or something will happen to it. Somebody will take a bite out of it. You obviously can't average over time. You can't, unless that's your question, which maybe it is, but probably it isn't. If something is changing over time, you don't want to average the times together. That just doesn't make any sense. So I'll leave that open to, like, how do you fix that? Do a time series is the answer. And we're going to look at that a little bit, but it can be a little hard to analyze. So we've learned a little bit about experimental design. I'll leave you kind of with this question from the very beginning of the slides. Is this pseudo-replication? Yay or nay? And also, does pseudo-replication matter? How do you get around it? And just consider that the circles, the black circles, those are the sources of, say, water that's going into all of those connected tanks. So knowing that is the pseudo-replication. And here's some more information about that kind of treatment. And then from Hurlburt and a couple of other folks actually have a newer version of this. I couldn't find it when I was digging through my slides and through my papers. These are the design types. And this is definitely not the be all end all. This is not the magnum opus of experimental design. This is just kind of like a simple version of looking at like what's good and what isn't. Here are what we can classify as good design in blue and less good design or poor design in red. So completely randomized, we like. Systemic, okay. Randomized block, also okay. The other ones I didn't even show you, they're not good designs. We don't like them. No replication seems really clear. That's bad. You'll see that one from the last slide, which, as you're going to learn, is pseudo-replicated, is randomized, but with interdependent replicates, because the water flows into all these tanks, then it flows into some sump, and then it comes back and it flows back into all these tanks. So in theory, all these tanks are actually connected, which is problematic. We can talk a little bit about these isolative experimental designs in class. I think that it's a lot to unpack. So what I'll also tell you in class is that the reason I didn't show you a better design on this slide is because there's another paper from Cornwall and Hurd from 2013 that shows a much more complicated version of what Hurlburt did, and it's specific to experimental tanks. So I want us to think about these experiments. We're going to do this together in class. We're going to talk about them. We're going to think about them, and we're going to try to make sense of what's good and what isn't. And it's probably going to take us more than 3 minutes, and that is totally, totally okay. But if you're interested in reading the paper, if you're missing class and you want to be part of these discussions, check out the Cornwall and Hurd 2013 paper on experimental design in marine sciences. And I think you'll learn a lot. I think it's specifically also about experimental design and ocean acidification experiments. Okay. So this is a table that shows you all the potential sources for confusion and error in an experiment. I'm just going to show you that it's here. There are many like it. You can check it out if you're interested. And then I'm going to kind of pitch that we will be talking through an experiment that I've done recently in Hartwell Sound that was pretty cool. I'll talk you through the details of it, but basically we were working with a community organization that's interested in oyster aquaculture and restoration to try to understand what kind of growing conditions might be best for oysters. And I will explain it to you in class because I want to show you some more photos and talk you through it because it will be cool. But I'm just telling you, like, we're going to talk about a real actual experiment and you're going to critique it pretty quickly. And then I'm going to pitch you another experiment, explain it in detail, and you are also going to critique it. The reason that we're thinking about this for our class is that the next couple of classes after this lecture, we're actually going to come in and I'm going to hand you and your group a paper. And you are going to critique it, look at the experimental design, try to tell me what kind of experimental design it is. And then you're going to just quickly present it to the class. Like, hey, we read this paper. This is what it was about. These are the things they found. Here's how they did the experiment. It went like this. It looked like this. It was great or not. And here's why. Remember that this is part of your experimental design project. You're going to do this case study thing 3 different times. So you're going to look at and try to analyze a paper with group 3 different times. You're also going to learn about 3 papers you didn't read from the other group. And then your job is going to be to do a detailed write-up of one of these explanations. You're going to mock up the experiment with a diagram. You're going to explain the design, maybe some positives, some negatives, some flaws, some biases. You're going to tell me whether you think it's doing the things it's supposed to do. And you're going to kind of demonstrate those 9 tenets of experimental design that you checked to see if they did all of those things and that you understand that those things are important. We're going to talk about that more in class. I'm hopeful that this unit will be really fun. And if it needs to take longer, like more days in our class, it can. So keep an open mind, be ready to explore experiments. There's a bunch of examples here in these slides. We're probably not going to get to them all. That's totally okay. I just want to spend more time having you think about and look at experimental designs. And a little bit less time worrying about like what every single term is called and whether like something is perfect or not. I want us to just think critically about whether the experiments will solve the problems that we're trying to have them solve or not. So we're going to be playing around a lot with experiments the next couple of weeks. I hope you're ready. See you in class.